Hey guys, welcome back to my channel. So, my name is Himani Agarwal, and today we are going to discuss the process of transcription in eukaryotes. So, in my past videos, we have discussed about this process, but in prokaryotes. Okay, so this process in eukaryotes is a little complicated one, and you have to understand this process very nicely. Okay, so we'll be discussing the process in detail, part by part. So let's start. So the content of this video is the RNA polymerases in eukaryotes, the promoters of different RNA polymerases, the general transcription factors, initiation and elongation, and finally termination of the process. Okay, so RNA polymerase in eukaryotes is different from what we have studied in case of prokaryotes. In prokaryotes, what we have seen is that there was only one RNA polymerase which was responsible for the transcription of all kind of RNAs. Remember, if you have not watched those two videos, please go and watch them. The description and the link is given in my description. So kindly watch them. In case of eukaryotes, the different kind of RNAs have been transcribed using different kind of RNA polymerases. In eukaryotes, three kinds of RNA polymerases are usually seen, which are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Okay, all of these different RNA polymerases have different kind of roles. Okay, so what is the difference between a typical prokaryotic RNA polymerase and an eukaryotic RNA polymerase? In prokaryotic RNA polymerase, what we saw was there was 5 units. Remember, alpha, 2 units of alpha, beta and beta prime along with the sigma factor, which makes the holoenzyme of RNA polymerase. But in case of eukaryotic RNA polymerases, each RNA polymerase consists of 12 subunits. So you can understand how complex the system is going to be. Okay. Another difference is that in prokaryotic RNA polymerase, we saw that it can directly go and bind to the promoter region and start with the process. It can start initiation, elongation, and then termination. But in case of eukaryotes, alone RNA polymerases are not enough to start a process of transcription of the genes. So there are different transcription factors, there are different enhancers, there are different activator proteins. This we will discuss slowly, part by part. Okay. Why different RNA polymerases are needed? As I've told you, because single RNA polymerase is not enough to transcribe all kind of RNAs. Okay. So let's see. What are the different kind of RNAs which we usually see? We see mRNAs, we see RNAs, we see tRNAs, snRNAs, and so on. So this is the general function of all different kinds of RNAs. Okay, so RNA polymerase 1 can synthesize 5.8S, 18S and 28S rRNA genes. What are these genes? If you remember, what are these genes? They are the large subunit of ribosomes in case of eukaryotes. Okay. RNA polymerase 2 is involved in all protein coding genes plus some snRNAs, okay, miRNAs, siRNAs, and small snRNAs. RNA polymerase 3 is responsible for tRNA genes, 5S RNA genes, and some snRNA genes. The most important RNA polymerase is RNA polymerase 2. Okay, this is the most important RNA polymerase in case of eukaryotes. You should know its function, its promoter, what are the different consensus sequence, how this RNA polymerase binds with different kinds of transcription factors, and how they initiate the process. RNA polymerase 2 is what is I'm going to discuss in detail. Okay, thank you. We'll be seeing the next chapter soon. Please like, share and subscribe to my channel and more informative videos. Stay tuned. Thank you.